Okay, this is part 2 of the Cancer Stem Cells lecture for Cell and Molecular Basis of Cancer. So, hopefully you've now read this paper by Kelly and you'll have looked through some of the points that I raise here, and you'll be able to draw your own conclusions about whether that work answers the questions in the way that the authors hope. So just as a bit of help to let you understand the pathways that are involved, here's a multipotent hematopoietic stem cell that can go to the common myeloid progenitor, common lymphoid progenitor, and if the cell goes down this lineage to the common lymphoid progenitor, the cells could differentiate down the B lymphocyte or T lymphocyte lineage, or maybe down the plasma cell lineage. B lymphocyte can give rise to B cell lymphoma, and the plasma cell to multiple myeloma. So the question you need to ask yourself is, at what point during this pathway do you think that the c-MYC gene will be switched on in that particular model? And it will be switched on at the point where the immunoglobulin heavy chain gene would be switched on, which probably represents a population of cells around about here. So basically, in that animal model, the hematopoietic stem cells would be normal because c-MYC would not be overexpressed in these cells, and probably not in these cells, and possibly not in these cells, but definitely would be expressed in these cells. So in that respect, the model is bypassing this stem cell population. However, it is thought that there are progenitor populations of cells which lurk around here on the diagram, which give rise to these. So it may well be that we're oversimplifying things by just looking at this stem cell population and ignoring the fact that there might be longer-lived progenitor populations around here that give rise to these cells. So they might be relevant to the study, but these are certainly not relevant to that study. Now, continuing that vein of thought, we're going to look at a prostate model, which I think possibly makes exactly the same mistakes. And this is the tramp mouse model of prostate cancer. This is a transgenic model, and the transgenic model is a result of a mouse which expresses the SV40 large T antigen under a probasin promoter. Now, this is a very prostate-specific promoter, and more specifically a promoter that is expressed in response to androgens, an androgen receptor, and the androgen receptor is highly expressed in prostate epithelial cells. So, probasin, androgen responsive promoter, large T antigen, is a viral oncoprotein that decreases p53 and PRB levels. Has a similar effect to human papillomavirus E6 and E7, but either way, we are creating cells which are devoid of PRB and p53 under an androgen responsive promoter. So only cells expressing high levels of the androgen receptor will be fully deficient in these two proteins. So mice have PRB and p53 decrease in androgen responsive cells. All of the cells in the mouse should have normal p53 and PRB levels. Now, the problem with this model is that the prostate tissue stem cells, although they are androgen responsive, they only very, very weakly positive for the androgen receptor. And the concern is here that the prostate stem cells themselves will not decrease PRB and p53, but the differentiated cells that come from those stem cells will. So therefore, there's a possibility that this model will only force transformation into cancer cells after a cell has differentiated from being a tissue stem cell. Almost identical to the stochastic model, in that in the stochastic model, cells differentiate, then acquire mutations, and then those mutations will propagate long-term growth. And that's effectively what's going on here. We are inducing p53 loss and PRB loss in more differentiated cells than stem cells. So with both of these models I've described, they both have the same caveats to whether the data that they generate is representative of human tumours or not. The real answer here is we don't really know whether these models are really good models or really poor models, and the jury is really out on this. Okay, so they're animal models studying cancer stem cells, and as I said, there are lots of controversies. Now what I'm going to move on to now is some in vitro models of cancer stem cells. Now if we're going to study cancer stem cells, we need some sort of cancer stem cell marker. And there are loads of markers out there and they differ for different cancers. We can look for cell surface panels, so alpha-2 beta-1 integrin, CD133, CD44. This defines a subpopulation of both breast and prostate cancer, tissue stem cells and cancer stem cells. The question is, if you take a tumour mass and you extract all of these cells from that tumour mass, are you getting cancer stem cells or are you just getting normal stem cells within the tumour mass? And this has been a problem of some of the earlier studies in this area. We can also look for other markers of stem cells such as ABC transporter channels. Now these ABC transporter channels are very good at effluxing toxins out of cells, and stem cells are very good at doing this. So we can use a dye called Hirsch 33342, and Hirsch can get into almost all normal cells, and this is just a two-way flow cytometry plot, and basically most cells are stained very strong on both axes, both Hirsch red and Hirsch blue. This is just nuclear bound and cytoplasmic, but the key point is most cells are up here and they represent that's the bulk population. There's this rare population here that is relatively weak for Hirsch staining. If we then add in verapamil, which can block these efflux channels, these cells that were weak for both in both Hirsch channels are no longer present because they are now part of this bulk population because they are Hirsch positive. So looking for the Hirsch, what we call the Hirsch side population, which is these cells, can identify a rare subpopulation of cells typically within cell cultures, and these may well represent the most stem cell-like cells within a population. Now, in most experimental systems, these do seem to represent stem cell-like cells, but they could also represent cells which have gained the ability to efflux this dye. Sometimes you can train cells to do this by culturing them with increasing amounts of low-dose chemotherapy, and they will increase their ABCB1 transporter levels and potentially potentially develop a Hirsch side population. Now this is nicely illustrated here. In this experiment we've got some lung cancer cells under light microscopy and under Hirsch staining, and you can see just how clear this is. On this diagram, if you look at this diagram and compare it to this one, these are the same cells. However, there's one cell here which doesn't appear on the fluorescent image, and that's because this cell is a Hirsch negative cell. All of these other cells have taken up the Hirsch dye. This cell has effluxed it, so this is part of the Hirsch side population. So this cell could well be a very, very interesting and important cell. And this cell represents one of these here, but then when you treat that with verapamil, all of these cells that are in this population end up in the high Hirsch stain population up here. This is exactly the same experiment or type of experiment as on this slide here. So it's all dealing with the Hirsch side population. We can also look for some more generic markers of stem cells within our tumour cell population. Again, this is all done in standard cell culture. One of these markers is aldehyde dehydrogenase. This is an enzyme present in many stem cell populations, including hematopoietic cells and mesenchymal stem cells. And some cancer cell lines contain rare ALDH1 populations. And this image is taken using an aldehyde dehydrogenase kit called Aldeflor. Basically, cells which are ALDH positive cleave a substrate and become fluorescently green. And what we found, this is one of our studies here, is that in prostate cancer cells there is indeed an ALDH1 population, and it is a rare population. However, not all ALDH1 positive cells are stem cells. Now, this is a mass of very rapidly proliferating prostate cancer cells, and in the middle there is a couple of green pixels in there, and there is one very bright green cell in there that's very difficult to see. We think that that is quite possibly a cancer stem cell like cell, but then equally there are other cells that form, often quite large, senescent, very unwell looking cells, or very spindly little colonies like this, which are also ALDH positive. So what we found is by selecting out all of the ALDH high cells and separating them from the ALDH low cells, we found that these, when plated out into new culture flasks, were far more proliferative than the ALDH low. So if we could isolate this cell here, put it into a new well, it would give rise to far more cells than if we took one of these cells. However, we also had a load of these cells which were non-proliferative as well, but overall we got more cells by selecting the ALDH high cells, suggesting that they might be very, very interesting. Furthermore, it wasn't just cell numbers, but when we plated these ALDH high cells into 60mm cell culture dishes at clonal density, that it gave rise to far more colonies than the ALDH negative cells, suggesting that they may well have a stem cell like or tumour initiating like population. And we refer to these cells as giving rise to the holoclone phenotype. Now, the holoclone phenotype, holoclones are, it's a terminology from stem cell biology. Holoclones are where cells have settled at clonal density and given rise to a very rapidly growing, aggressive looking, rapidly growing colony where the cells pile up on top of each other. So they're growing really fast here. There's holoclones, mericlones and paraclones. Paraclones are cells which are terminally differentiated and have limited replicative potential. Mericlones are cells that seem to settle and give rise to a decent sized colony, but they're nothing like as fast growing as holoclones. And importantly, if you take this population here that gives rise to this large aggressive colony, pick off these cells and plate them out into a clean, a new cell culture well, these will give rise to more holoclones, some mericlones and some paraclones. Whereas if you take mericlones and put them into a new dish, they'll only give rise to more mericlones and more paraclones, and if you pick off these cells here and put them into a new well, they tend to not grow at all. So these seem to contain cells which have a stem cell like phenotype. These probably don't, and these definitely don't. So this was a study that we did a few years ago now, identifying this cell population within PC3 prostate cancer cells. And the one thing that we can be very sure about here is that only a minority of cells in a cell culture flask can give rise to a large aggressive colony like this, and in our hands it was about 5% of the colonies that were formed were able to give rise to these large aggressive colonies, and these seem to dominate and propagate the cell line. These two types of cells giving rise to these two types of colonies get rapidly outgrown. Okay, there's another marker that we can use to look at stem cell like cells, and I've been talking about stem cells, potentially giving rise to more cells here. So stem cell like cells being more proliferative, but the stem cells themselves are very non-proliferative. They don't divide very often. It's their progeny that divide very, very rapidly. So in there is one green cell. That cell is probably non-dividing. It's divided once or twice, but its progeny have given rise to this, while that cell just sits there and doesn't do anything. We can study that, and that idea of a cell giving rise to proliferative and non-proliferative progeny in the non-proliferative cells, we call them quiescent cells. And this is an experiment that we perform fairly routinely in our lab, which is to grow cells that have been stained with a dye called PKH26, and PKH26 stains the lipids red, and the only way the cell can get rid of it is by cell division, so that its daughter cells are half as red as it was, and then as cells divide more and more and more, they become less and less red. And what we find that if you start off with a population of very red cells and follow them over the course of a few weeks, you end up with a population of cells which have lost all of their red colour because they've been dividing like every 24 hours, and then you've got other cells which have not divided at all within that time period. What we do is we separate those cells out by flow cytometry and flow-aided cell sorting, and separate them out into different wells and then study them in isolation. And this is what we've done here. Started off with some very red stained cells, waited for over 2 weeks until we've got a rare population of red cells, separated them out. These are the PKH26 high, these are PKH26 low, and then we study how they respond to drugs, and they behave very, very differently. So these are some cells from our multiple myeloma studies that we've recently done, and these are unsorted cells, and they've been treated with a drug combination, TRAIL and BIX01294. That's a histone methyltransferase inhibitor. It doesn't really matter what the drug combination is, but what you can see is these cells, they've been stained, well, they stain red because these are the unsorted cells. Red is PKH26, green is caspase 3, so apoptosis, and the blue is Hirsch 33342, which we're using for nuclear morphology, and the condensed bright cells are apoptotic. And the overwhelming majority of these cells in here are apoptotic. And this is just the DAPI channel, also just the blue channel in this diagram. Now when we look at the PKH low cells in isolation, every single one of those cells is apoptotic. However, when we look at the PKH high cells in isolation, by looking at this channel here, you can see that only an extreme minority of them have got condensed nuclei and are apoptotic. So that suggests to us that the PKH26 high cells that we've selected out are almost completely drug resistant. The green ones are apoptotic. That green co-localizes with the condensed nuclei of the Hirsch staining in this channel, and all of the red cells are pretty much normal in the DAPI channel, which is the Hirsch channel, same thing. So it's saying that these non-proliferative cells are completely drug resistant. Now what is really interesting about this, and I've not shown the data for this, but because this is multiple myelomas are normally CD138 positive, and we know that the CD138 negative population is very, very drug resistant and is crucial for initiating relapse from standard therapies. When we looked at our PKH high cells, they were all CD138 negative. So that suggests that we are pulling out our cancer stem cell-like, certainly drug resistant, certainly the cell type that induces treatment failure. We are pulling out those cells by selecting for the PKH high population. So we're fairly confident that we've got a very interesting subpopulation of cells here that is relevant to disease. Okay, the final model of potential cancer stem cells I'm going to show you, and this is not my data, and this is looking at stem cell populations which might have a low proteasome activity. So the proteasome is an organelle that degrades proteins in a very targeted manner. You will have covered this in therapeutic targeting in cancer module where I talked about the drug Velcade. So the proteasome, cancer stem cells or the stem cell population seem to have a very, very low level of proteasome activity, and we can visualize that in this model. Although this is not my data, we've generated very, very similar data to this. It's just that I didn't have quite a nice figure to hand. What has been done here, and we've done exactly the same thing using a green fluorescent protein linked to mouse orthonine decarboxylase protein, and this mouse orthonine decarboxylase is rapidly degraded by the proteasome. So if you've got GFP linked to a protein that is degraded by the proteasome, your GFP gets degraded by the proteasome as well. And if we grow cells in standard monolayer culture, you don't see any green colour. And we've seen exactly the same thing in our model systems. If you grow cells as spheres, so grow the cells on low adhesion cell culture plates and induce 3D cell culture, which I'm going to talk about in my next lecture, what you find is that these cells tend to accumulate green fluorescent protein because their proteasome levels are very low, and that is characteristic of stem cells. And the induction of spheroid growth is very closely linked to cancer stem cell behaviour. In our lab we induce spheroid growth like this by two ways. One, by not allowing the cells to stick to the plastic, and then they'll undergo spheroid growth and accumulate cancer stem cell markers. And the other way we induce spheroids is to grow them on low dose doxorubicin, and then the surviving cells efflux the doxorubicin, and they, in exactly the same manner that they efflux Hirsch, and you end up with cells which also spontaneously form spheres. So the presence of a proteasome low phenotype correlates with accumulation of GFP fused with mouse orthonine decarboxylase, so that tells you that these cells here are proteasome low. So, in summary of everything I've covered, there is no single marker that can confirm that some cells are cancer stem cells, but there are markers that can show that cells contain stem cell-like features, such as ALDH1, and that also selects for holoclones, which is a stem cell-like phenotype. We know that ALDH1 positive and Hirsch side population positive cells coexist, which is strong evidence that these are stem cell-like. We know that detecting usually 2 or 3 different stem cell related markers, so alpha beta 1, CD133 and CD44 positively identifies the stem cell population in prostate tissue and in prostate cancer cell cultures. We certainly think this is very, very relevant to prostate stem cell biology and may have some relevance to propagation of prostate cancers themselves. Different tumour types have different panels of markers that positively identify their stem cell population, so no single panel works for all tumours. And then finally we've got the PKH high quiescent phenotype. That in our hands co-localizes with the proteasome low phenotype, so we think that this is a really good marker of certainly a stem cell like phenotype, especially when the PKH high phenotype proteasome low phenotype correlates with, in our hands in multiple myeloma, CD138 negative, which are the drug refractory cells, which are the ones that survive Velcade treatment in multiple myeloma. So we're very interested in this population of cells. They might not be a classic cancer stem cell population, but they are certainly disease and therapy relevant cells. So in summary, cancer stem cells are incredibly difficult to define. There's no universal specific marker. You'll struggle to find 2 cancer biologists who completely agree on everything to do with cancer stem cells and their markers, but we certainly know that cancer stem cell markers differ between different tumour types. Within tumours there's definitely a hierarchy of cells. My favourite definition of a cancer stem cell is the cell that is at the pinnacle of the neoplastic hierarchy. That basically just means it's the cell that's the most primitive. However, a primitive trait such as that may also be plastic, as it undergo plasticity. So a cell that on one day is differentiated, the next day may be the most primitive by undergoing phenotypic plasticity. So phenotypes within cancer are not fixed, and that means that targeting cancers on the basis of a single phenotype may mean that you are constantly missing the cell that is propagating that tumour. It's also possible that all cells are potential cancer stem cells if you put them in the right environment. So, unfortunately I can't give you any concrete answers on this lecture. All I can tell you is the whole field is very controversial. And the most important thing is that you understand the in vitro methods to study cancer stem cells, the in vivo methods to study cancer stem cells, and you are aware of the potential caveats to the data that is obtained. So I think that is the key thing to understand is understand how the methods work and what they are trying to show. However, you need to understand the limitations of all of those methods. Okay, I think that's probably enough.